We continue our discussion on the excretory system of animals. Now we will focus on the excretory organs of the human body. So first is we look into the kidneys, which is a primary filtering organ of the animal body of the human body. So let's take a closer look on the anatomy of the kidneys. So the kidneys are a pair of structures that are found at the back of the abdomen, behind or outside the peritoneum. So that's why we term it, or there's a specific adjective to describe its anatomical location. So the kidneys are termed as or described as retroperitoneal because they're found at the back of the abdomen behind the peritoneum. Okay, and there are specific entrance point or section called the hilum or the hilus, where the structures, blood such as blood vessels, the ureter, enter in order for the kidneys to receive blood supply that needs to be filtered. Okay, so the major blood vessels related to the kidneys are the abdominal aorta and the inferior vena cava, which has branches entering both kidneys. No? So, uh, the primary function of the kidney is to regulate um, the the parameters of the human blood, the pH, the pressure, its concentration in terms of solute, ionic composition, no? And in the kidneys or via the kidneys, filtration of waste material and foreign substances take place, and those filtered out wastes or unwanted materials are then secreted or excreted via the urine. Okay, and in terms of the development of kidneys throughout the lifespan of the human animal, no? So during embryonic development, the kidneys start as structures called the phronephros, which are temporary. They're the embryonic kidneys, and mostly we expect them to degenerate to eventually be replaced by a transitional structure called the mesonephros, which serves as the adult kidney, okay, in most vertebrates. And in some, it can serve as the functional kidney of embryonic amniotes, such as reptiles, birds, and mammals. And we have eventually maturing to metanephros. The metanephros are the adult state or the adult kidney. No? In terms of its appearance, the kidneys are dark red in color. They're kidney bean shaped organs, and their size about the size of our fist. So when you close your palm, or when you make a fist, the size of your fist is about the size of your kidneys, and it's covered with fat and a capsule called the renal capsule, covers the organ, covers the kidneys. Okay. Internally, if we look into, like, if we cut through the kidney, what are we going to see? So if we cut longitudinally, okay, this is how the internal structure of the kidneys look like. So there is the renal cortex, the renal medulla. So the renal cortex is the outside structure. It's the you can say you can compare it to the what do you call this, the covering of a fruit. Okay, the first layer of flesh that you see if you peel a fruit. So it's similar to that. So it's a cortical region which is surround which is um, um, uh, proximal to the surface, so it's more superficial compared to the medulla. So the medulla, okay, is of course is a structure that is close to the pelvic region of the kidney where concentration of urine happens. Okay, so the medulla below the renal cortical region. Okay, um, the renal pelvis is uh, a cavity that serves as a catch site, a catch basin where urine is collected, okay, and eventually urine is transported from the renal pelvis down to the ureter, where it can be expelled later on during urination. No? Microscopically, okay, if we uh, uh, prepare tissue sections of the kidney tissue, we'll be able to observe key structures, microscopic structures, that are actually the functional unit for filtration, no? For waste filtration. And these are the nephrons here. So the picture here, you can see the one I'm highlighting. Sorry. Oops. It's not working. Hmm. Oops. I don't know. It's not working. Anyway, so the microscopic structure, the microscopic major functional units of the kidneys, the nephrons, you can appreciate here are associated to both the cortical and the medullary region of the kidney. So the cortical, the outer layer, and the inner layer. Okay, so we divide the structure, the nephron, into different parts. So there is the renal corpuscle. This one is sac here. It's a renal corpuscle, and then there is the tubular part, which are all tubules. Okay, right. In terms of types, we have two types of nephrons. So we have the cortical nephron. Okay, cortical nephrons are are cortical nephrons have shorter Henle loop. So they have shorter nephron loop compared to juxtamedullary nephrons, which have a longer Henle loop. Okay. Now, looking into the different parts of the nephron, okay, we'll discuss them one by one, but to enumerate them, so we have the renal corpuscle, which is made up of um, primarily convoluted or a ball of capillaries where blood passes through. This ball of capillaries is where blood passes through, and it's divided into two types, or it has two major portals, an entry and an exit portal. So there is an afferent blood vessel, this one I'm highlighting, which allows the entry of blood towards the glomerular capsule, and an efferent vessel, this other one here. Okay, let's delete all the other highlights. Okay, so we have the afferent vessel allowing blood entry, this one highlighted in green, and we have the efferent vessel. Okay, sorry, this one highlighted in yellow, which allows the exit of filtered blood. Okay? Then there is connected to the glomerular capsule, to the renal corpuscle, are the tubules, the renal tubules. So we have the first part called the proximal convoluted tubule, the PCT, right? Which is termed as such because it's nearest to the renal corpuscle. And then there is the loop of Henle, this one I'm highlighting, okay, which connects the proximal convoluted tubule with the distal convoluted tubule. Okay, so we have the renal corpuscle, the proximal convoluted tubule, the loop of Henle, and then the distal convoluted tubule. Right? Then later on, the continuation of the tubules allow for the transport of material towards the collecting ducts that will open to the renal pelvis, right? Okay. More on the types of nephrons. So juxtamedullary they're called juxtamedullary because a good portion of it is actually present in both in a good portion of the nephron is present in both the cortical layer and the medullary layer of the kidneys. While for cortical nephrons, they're called as such because majority of its parts are found in the cortical layer. Okay. More about the loop of Henle. So there is a descending loop or descending limb and an ascending limb. Okay. So the descending limb connects the PCT to the loop of Henle, while the ascending limb connects the loop of Henle to the distal convoluted tubule. Okay. Yeah. Right. So okay. So discuss this. Yeah. So more on the more on the anatomy of the corpuscle and the tubule. So if you look into the renal corpuscle, right? So it looks like this. Let's try to blow up the picture here. Oops. Oop. Okay. Mm-hmm. All right. Let's try to zoom in. Okay. Mm-hmm. So the renal corpuscle, okay, looks like this. All right. So you can see the afferent vessel, the afferent arteriole, blood entry, efferent, blood exits. And if you, though it's not really evident here, okay, afferent arteriole is actually larger in diameter compared to the efferent arteriole, allowing for entry of more blood to be filtered by the capsule. And efferent arteriole has smaller diameter, increasing the pressure, okay, within the blood pressure within the renal corpuscle. And then we have the within the glomerular capsule, okay, so we can appreciate the presence of the glomerular layer of filtration, which is composed of the capsular epithelium of the Bowman's capsule, podocytes, and the endothelial epithelium, no, the endothelial layer of the capillaries, right? So um, within the glomerular layer of filtration, or the glomerular layer of filtration facilitates the transport of material, the filtration of material from the blood. Okay? Then we look into the renal tubule. So I've already discussed the major parts. So we have the PCT, the loop of Henle, okay, the DCT, and the collecting tubules that will divert the material collected towards the bladder. Okay. So, if you notice, no, across the, or I mean, surrounding the tubules, so there are associated capillaries, blood vessels that are also found. So the capillaries are not only confined within the Bowman's capsule, within the renal corpuscle. Okay, so there are blood vessels, capillaries that surround the tubules for the transport of material, for transport in and out of the blood, okay, from or to the tubules, because um, the as the filtered material is transported from the Bowman's capsule to the renal tubule, a number of things actually happen. No? Not only that filtered material is not simply transported. No? So there are several things that happen. Reabsorption and secretion are two of those key activities that occur within the renal tubule, and we'll discuss each of them. Okay. So I've mentioned, discussed this already. Okay, so here, this slide. So this one shows us a schematic of what materials are transported or reabsorbed, okay, uh, within the tubular region of the tubular region of nephrons, okay. So within the Bowman's capsule or exiting the Bowman's capsule, water, amino acids, sugars, urea, ions such as sodium, potassium, and chlorine, they're all transported. Then urea salts, urea and nitrogenous wastes are kept um, within the filtered fluid, okay, in the uh, proximal convoluted tubule, tubular reabsorption takes place. So we want, as much as possible, we want to maintain or regain sugars that are filtered out, sodium, amino acids that might still be needed, okay, and some extra water if it's not to, or if the body still requires it to maintain physiological conditions. So those reabsorption takes place in the proximal convoluted tubule. Okay, then in the lubohenle, water is reabsorbed. Additional ions such as sodium and chlorine are also reabsorbed back to the blood. Okay, then in the distal convoluted tubule, tubular secretion takes place. So if there are unwanted drugs, there are ammonia, hydrogen, and potassium that are not needed anymore. They are secreted from the capillaries surrounding the tubular region, the renal tubule, to the distal tubule, okay, or to the fluid that now, the filtrate that now travels across the distal convoluted tubule. So you'll see that urea, of course, urea, nitrogenous wastes, they're not reabsorbed by the body. And hydrogen ions are also secreted or filtered out, or they are secreted out, okay, from the blood towards the urine, okay. And then all those concentrated material that are filtered out of clean blood are now going to be concentrated as urine. Okay. I think I just, but this is just a cartoon representation of what's happening. But yeah, you have to take note of the things that, or the components that are either reabsorbed or secreted, and which structures of the nephron are responsible, or which structures are the sites for these reabsorption and secretion processes. Oh. Okay, what else? Okay, so now that we have a concentrated urine, that's with all the wastes, excess ions, excess water, that urine will be diverted to the ureter, okay? Then eventually to the bladder, which serves as a temporary storage for urine. And when the animal is red is or the bladder is full, the animal or the human urinates to secrete or to excrete the waste material via the opening urethra. Okay, so that ends our discussion on the animal excretory system.